timed like that before? I haven't. Good evening and welcome. My name's Stephanie Smale. I'm a journalist with the ABC's Radio National Breakfast program. I would like to begin by acknowledging the Gadigal people of the Eora Nation as the traditional owners of the land that we are gathered on tonight, and to pay my respects to elders past and present. And I'd like to extend that respect to any Aboriginal and Torres Strait Islanders here with us tonight. As we could see from the spectacular images playing on the screens earlier, viruses can be surprisingly beautiful close up, but we know they're also dangerous, potentially deadly in fact. The CSIRO is at the forefront of the battle against infectious diseases. From the flu some of us will be unlucky enough to deal with this winter, to the Hendra virus and even Ebola. The nature of the work means the scientists developing ways to protect the public, such as life-saving vaccines, are often toiling away in highly secure labs. So tonight we have the rare and exciting opportunity to take a peek behind the scenes and to hear about the world-leading work being done to battle these potentially deadly diseases. We'll hear about why the study of animals is crucial, why vaccines are so important, and how data visualisation, like these amazing images that are part of Vivid, is helping in biomedicine. It's my pleasure to introduce our first speaker of the evening. Sean O'Donoghue is a Senior Principal Research Scientist in CSIRO's Data 61 team and also at the Garvan Institute of Medical Research. Sean's expertise is in data visualisation, and he is one of the very talented biomedical animators who put together CSIRO's Vivid Light installation on George Street in the Rocks, which is directly behind us. It's fabulous. Welcome.
So if you go down behind the building, you'll see this is a short scene from the animation that we're playing, projected onto the old police station and bank that's just there in the rocks. And actually the subject of this, the hero if you like, is a virus. That's what one virus looks like. And part of the process of what I'm trying to show is how much we know about some viruses. We know actually exquisite detail in some cases. And most of viruses, like most of us, are made up of proteins actually. And not many people realize that the viruses that infect us, we actually make them. We actually build the viruses that then infect us and others. Because what viruses do, of course, is get into our DNA and force us to make copies of themselves. They're too small, they're too simple, they can't replicate themselves. And if I go on to the next slide. So actually, a couple of years ago, I'm going to tell you a little piece of science, not too much, but I want to take you a little bit of a journey. So I was really fortunate to be leading a team, and we did essentially like a survey of everything we know about protein structures, not just viruses, but bacteria and everything. And what we did is we said, well, let's look at a typical protein. This is a typical protein here. And in some parts of the protein, we know exactly the molecular organization of that part of the protein. We know almost at the level of the atom exactly how it folds into 3 dimensions, and that means we can say a lot about how it works, how it functions. And there are other parts of the protein that we actually don't know. We've never experimentally observed them, but because they're similar to things, whoops, can we go back one? Okay. No, never mind. There are other parts of the protein that we don't know, we never observe, but we can actually map them onto something we do know because we can recognize the similarity. And we did this systematic survey, taking everything that we know, mapping them onto every protein sequence that we know, and we found that we could predict quite a lot of it, but there were these regions in the middle that we knew that we couldn't predict. And we realized with a bit of a shock that it was a stronger statement that we don't know the organization of the protein there. Actually, we know that those dark regions, we called them, aren't similar to anything that we've ever seen the structure of. So we really have no idea what's going on in that part of the protein. And then we mapped that across all of animals, plants. We got this distribution where the actual amount that we really know experimentally is maybe it's 3 to 4%. And based on that 3 or 4% that we've observed somehow in these experimental systems, we can model about half of it, and that leaves about half of it completely unknown. And of the half that's unknown in animals and plants, a third of it is actually the entire protein is totally unknown. We call those dark proteins. And then the interesting thing was when we looked across the different areas of life, I think I missed a slide there, but okay. Maybe, well, somehow didn't come across in the presentation, but virus is actually basically very similar to animals and plants. About half of the viral structure, it's kind of frustrating. Half, I can tell you pretty much exactly, and even tell you how exactly. I can tell you how it's organized down to molecular detail. The other half, I have absolutely no idea. That's both true for animals, plants, and also true for viruses as well. So we have a lot of mystery, a lot left to explore in biology. And so I just wanted to make you aware of some resources that my team's been creating. We've been creating a series of biomedical animations that use these models to create molecular stories that can communicate various parts of biomedical science in really interesting detail. These are all on YouTube. It's a website you can go and check out. And with that, I'm just going to make you aware of some vivid activities we're doing. So we're running a course in how to visualize data, how to make these kind of molecular animations and other kinds of animations on Thursday. And on Friday, back in this room, Janet Iwasa, who's joining us tonight, is going to be, and Martin Krasinski, are going to be talking about application of data visualization to a really wide range of topics. So with that, I'm going to finish up and hand over to my colleagues.
Our next speaker is Rebecca Johnson, who's a postgraduate student in CSIRO's Health and Biosecurity team. She's based at the Australian Animal Health Laboratory in Geelong in Victoria. The lab is one of the most secure facilities in the world, certified for working on infectious viruses. Rebecca Johnson focuses on the discovery and characterisation of new viruses from Australian fruit bats to find these viruses before they cause the next outbreak. Welcome, Rebecca.
All right, so I'm going to assume that you have all heard of Ebola virus. So right now, an outbreak is actually taking place in the Democratic Republic of Congo, where over 50 people have already been infected and 25 people have died as a result of this outbreak. What about Nipah virus? There's also an outbreak of Nipah virus taking place at the moment in India, and so far 14 people have died in the last few weeks. Or even SARS coronavirus, which caused respiratory disease and death all over the world in the early 2000s. Now, all of these viruses have something in common. Before they cause disease in humans, they are all thought to have originated in bats. Bats carry a huge number of different viruses. In fact, they carry more viruses with the potential to infect humans than any other mammal. What this means is that we need to look at bats to try and find these new viruses before outbreaks even happen. What's really interesting is that they can carry these viruses without even getting sick. There's many reasons as to why these bats carry so many viruses. One of the potential reasons is because bats have a completely different immune system to us. Part of the immune system of bats is always switched on and ready to go, whereas our immune system needs a signal before it can get going. What this means is by just looking at this bat, we have no idea what viruses it's carrying. Does it have Ebola virus? Does it have Nipah virus? Or does it have some other unknown virus that's just waiting for the right time, for the right environmental conditions, before it moves into animals or even into humans and starts to cause disease? One of the problems we face is that bats actually carry hundreds of these unknown viruses. So instead of waiting for someone to get sick before we try and investigate it, what I'm trying to do, and what many other people around the world are trying to do, is find these viruses while they're still just in bats. In particular, I've been looking at Australian fruit bats, like the grey-headed flying fox right here. And because we don't really want to disturb the bats, we actually collect the bat urine to try and find these viruses. And as well as being very cute, these bats are also very important to our environment. So we don't want to get rid of them, but we do want to try and stay away from them and work out what viruses they're carrying. So to get the virus from the bat into my lab, I collect the bat urine and then try and see what viruses are present in there. And then I try and work out if these viruses are actually able to cause an outbreak. So can they infect people? Can they cause disease? Can they maybe transmit from person to person? Once we understand more about these viruses, we can use this information to then maybe develop diagnostic tools. So we have an early warning system already set up and ready to go. We might even be able to develop treatments before an outbreak happens. Because we don't really know much about these viruses before we start working on them, I do all my work at the Australian Animal Health Laboratory, which, as Steph said, is a high containment facility. So it allows us to work on these viruses without the risk of people getting infected. And as well as all the amazing engineering that goes into containing these viruses, one of the things that we have to do is we have to shower and wash our hair every time we leave the facility. So we're scientists with very clean hair. Luckily, the viruses that I'm working on are unlikely to cause a deadly disease in humans. But if they were, at ARL I have the ability to move them into an even higher containment section of the facility. So there we're even more protected from the viruses because we're essentially working in an inflatable plastic suit. It has our own air supply, which is the blue tube, and it completely seals us off from any viruses that we're working on. And this is really exciting because there aren't many of these facilities around in the world, and it allows us to work on viruses like Nipah virus and like Ebola virus. So finding the virus that will cause the next outbreak is obviously a massive job. In my PhD of around 3 years so far, I can tell you a lot of information, but only on 2 viruses from just one Australian bat species. But there are over 100 species of bats alone, let alone other mammals or birds or mosquitoes, and all of these have their own set of new viruses that need to be discovered. So that's why it's not just a job for me and my PhD, or even for the CSIRO, but people all over the world are working on finding these new viruses and then preventing the next outbreak from happening.
Our next speaker is Andrew Bean, who's a research team leader for disease prevention and detection in the Health and Biosecurity team. And he's also based at the Australian Animal Health Laboratory in Victoria. Andrew is an expert in the immune system, and he's working to develop better treatments and vaccines and improve disease diagnosis in humans and animals. Welcome, Andrew.
Thanks very much. So this evening I'll talk a little bit about flu. You know, this year marks the 100th anniversary of Spanish flu pandemic, which killed over 50 million people, and that's an absolute tragedy. What was really very bad about this flu was that it tended to attack people who were in the middle age and people who appeared quite healthy and caused their deaths. So it's been 100 years since then, and there's still so many things that we still don't understand about influenza. And even seasonal flu that we see every year, it causes 500,000 people to die each year. Again, in the current world we live in, that's an absolute problem and something that we need to do something about. Even if you think about last year for us here in Australia, it was the worst flu season that we had on record. Over 200,000 people were diagnosed with influenza, and we saw a lot of deaths last year as well. So as much as we know a lot about influenza now, there are still so many things that we need to do to help improve our quality of life. So right now we're just about to start what we would say is flu season. And there are 2 different flu seasons, as you might imagine, in the world. In the southern hemisphere, we have our current flu season, which usually runs from around June until October. And then in the northern hemisphere, they have theirs at a different time of year. For them, it's more like, the end of the year, November through till about March. And so you always need to be aware that if you're going on holidays in the Northern Hemisphere and you're going in February, be careful. You should get flu vaccinated for their flu season. But obviously for us here now, our flu season is when you should get vaccinated here. Now, what about the flu virus? You've probably heard a lot about it, and you hear it by a lot of different names. There are 4 different types of flu viruses. There's A viruses, which are usually in animals and also in humans. There are the B viruses, which infect pretty much just us. There are C viruses, which are really quite mild and don't seem to be associated with any, any seasonal flus, that type of thing. And then there are the new D viruses, which really only appear in cattle and, sometimes in pigs. But again, you probably have heard people talk about A flu virus or B flu virus, because particularly last year, one of the A viruses was really serious. But that's what they're talking about when they talk about those sorts of different things, the viruses there. And just a reminder, there are no E viruses yet. Now, flu symptoms. Lots of people say, oh yes, I had the flu. Yeah, how many people have you heard say that? And you know that they probably really only had a cold. Because if they were standing up talking to you and telling you they've got the flu, they probably haven't got it. So there are many viruses that can cause colds, probably about 200 different types of viruses that can cause a cold. And that's why even in the winter, you probably get a couple of different colds across that winter season, because there are a number of different viruses that cause it. But when it comes to flu, there are many other symptoms that are there that differentiate it from a cold. A cold really is just something that's quite, I guess we'd say it was quite mild by comparison. Flu has many other things, and that's like muscle aches, fevers, chills, those types of things. And can even give you stomach problems, can even end up with diarrhea. So it's a far more severe infection, and it certainly has a lot more oomph. Now, flu obviously has these kinds of symptoms, and there are many treatments that are available for flu, and it's really important that people take the right approaches to it, because when you are infected with flu, from about a day before you're showing signs, you can infect others, and right through to even about a week afterwards, you still can infect other people. So we don't say soldier on anymore, we say stay home, keep it at home. So like I said, the important solutions that we have, and I think that most people would agree that for influenza, the main approach that's going to be most effective for us is vaccination. The flu vaccines were first developed in about the late 1940s right through the 1950s. It wasn't until we understood that flu was caused by a virus that we could even consider what we were going to do about how to treat it. And that really wasn't until, early in this century. So flu vaccines weren't developed for quite some time. And then it was really important to work out a way that we could grow and make these flu vaccines. And fortunately, in the 1930s, they were able to identify that the virus could be grown in eggs. And so flu vaccines are produced in eggs, and you've probably heard that before. So it's really important, and it's a critical process, the development of flu vaccines. And as I say, an Australian played a really important part in the role of developing flu vaccines in eggs. Macfarlane Burnett, who was from the Walter and Eliza Hall Institute in Victoria, he played an early role in that development, that idea of being able to grow up the vaccine and be able to prepare it. And in about 1945-ish, or around that time frame, was the first time we saw mass vaccinations occur, and it started with the US Army, and then shortly after that, it was offered to people. So vaccination really is one way forward for protecting the population. So every year, the WHO and the CDC in America, they're given the task of having to survey what flu viruses are going around so we know what to do for next year. And this is one of the main difficulties that we have with vaccines, and sometimes why we probably don't get the best protection that we would love to have with each vaccine, because it's very much about having a look at what's going on. As I said earlier, there are 2 flu vaccine seasons. So the WHO looks at what's going on in the other season, and from that tries to predict what might happen in our following season. And in that brief time that there is between the two seasons, companies have to get out there and rapidly produce that vaccine, get it up to high levels so it's ready to go for when we need it. So vaccine manufacturers are under considerable pressures to try and get things done. And that's probably one of the reasons why we often hear, from year to year that we're running low on flu shots and those sorts of things, because it's a very tough, tight timeline to get these things done. And a lot more work needs to be done and is being done, particularly by CSIRO, around developing new processes for us to be able to produce the vaccine, to try and get it into much higher levels so that it can get out there and it can get out there faster and provide that life-saving protection to people. Okay, so I just finished off by putting up this picture here. This is a picture of my brother. 1 year, he forgot to go and get his flu vaccine. And he had to go to hospital and he was in intensive care for a couple of days and then had to be put in isolation after that. Here he is the other night when I was out with him, and I think he's much happier there. But let me tell you that he told me to tell you, you should go and get vaccinated. You don't want to go and get flu. So thanks very much.
Our next speaker is Kathy Foley, who's the Deputy Director of CSIRO's manufacturing team. In this role, Kathy leads a science research team that is helping to treat and prevent diseases, benefiting millions of people around the globe. Welcome, Kathy.
So you've heard some pretty gory things about how there's all these dreaded diseases around ready to take over. All is not lost because we at CSIRO are also working at how to make sure that you are able to get medicines and things and treatments that allow you to survive it. So that's the bit I am working on. I'm a physicist, and so hearing all about this stuff is really fantastic. But I have the great pleasure of working with a whole lot of people whose job it is to look at how, oh, this is going too fast, isn't it? How we can take chemistry and biology and also physical devices to be able to create products that help you with your health and well-being. So I don't know about you, but I always thought that polymers were things like nylons and plastic lunch boxes and things like that, but our whole body is made up of a whole lot of polymers. And for a very long time, there was this idea that you couldn't do anything to actually control them. So you'd make a cup of polymer and you'd have some which were long and some which were short, and they were never very pure. So one of the things that was really important to be able to make good medicines was to be able to control how these polymers are formed, because we're actually all made of a whole lot of polymers. And so what you're looking at there is a whole swag of them. And something that's been really amazing is some scientists in CSIRO have actually cracked that problem, which actually required the chemistry textbooks to be thrown out because they thought that this was absolutely impossible. And they developed this technique which is called raft. And it's ability to put in an extra element that allows you to get in there and control the length of polymers. So that's something that's really important. It's a really basic part of science, but it allows us to be able to control the chemistry to make the medicine that makes us better. So the other thing that's interesting with this is you can also control polymers to have all sorts of different shapes in order to make sure that the way that the chemical that they've developed is able to interact as best as possible within your body. And the thing that's really, oh dear, it's really got a life of its own here, hasn't it? And so in this is looking at some science pictures that at the top, it's looking at getting an X-ray, and X-rays were actually invented in Australia by, well not invented, but discovered with the ability to do X-ray crystallography by the Bragg father and son in South Australia. Oh dear, that's got I think it must be on an auto thing. And what they did was you're able to put a chemical in front of an X-ray beam, and each one has its own unique structure, which is shown at the top there. What you're seeing there is, and we'll see if we can stay there long enough, is you can vary the different shapes by playing around with the chemistry, measure it, and be able to, in this case, get the variation into the surface area. Because surface areas are good because that's how your body can interact with the chemical. Whoops, and so here we're looking at the idea then, how do we get to know what are these viruses, what are these polymers, of which viruses are, and what are they, what are the chemicals made of, and how can we actually study them so we know how to create a drug to fight them. So this is a machine here that goes through, and you can see a little picture there, which I've got a better picture here. Oh, no, it's not going to go at all. Here we go. And in the top corner, you can see they can go through and pull out these proteins or these polymers, which are very small, and be then able to go through the process of doing that X-ray crystallography to be able to understand its structure, and then go through to a point where they can get what looks like a nice wrapping ribbon, which actually is a way of representing the chemicals that are present. And from that, you're able to study it and get to know what's there and find out, can we create a drug or a medicine that can interact with something that will help us to fight it. So here's one example. What you're looking at there is a classic flu virus. You've heard that flu changes all the time. But one of the things that they asked was, is it possible to actually get the bit in the flu virus that doesn't change? And so therefore, once you've got that, can you create a drug so that even if you didn't get your vaccination and you do get the flu, can you take a medicine that is an antiviral that kills it off? So the bit that's marked there in orange was some fastidious work that was done, being able to find out which bits of all flu viruses do not change. And then what they did was go through, and some other people in, I think, University of Queensland went through and found that there was a particular chemical which was an inhibitor that was able to get into that little bit that was which stays the same in all drugs and create an antiviral drug. And so when you get the flu, you can go and get Relenza, which is an Australian invented drug. It's an antiviral. It's got very low drug resistance, which is good compared to some of the other antivirals. It has one little problem though, and that is you have to sniff it. And that's a whole other thing. You can ask me a question as to why did we do that later. But it's really interesting is that people in Western countries think sniffing goes with something else, which we won't go into here. And so it's not a preferred way. And Tamiflu, which is another antiviral drug, is the preferred one because it's taken orally. So that's drugs, okay? So that's sort of how they're developed for being able to deal with viruses. But I thought I'd also let you know there's other things we do too in manufacturing in order to make sure that we've got things that help us with our health and well-being. So what you're looking at there is the green whistle, and you might have seen it in Bondi Lifesaver or whatever. It's an analgesic which is able to be breathed in through this green whistle. And then when you get a bit dopey because it makes you groggy, it takes away chronic acute pain. You drop because it drops out of your mouth because you get a little bit drowsy, and you can't overdose on it. It means it can be used by paramedics, and it's actually reduced the pains of millions and millions of people around the world. The thing that's really interesting with this is we helped develop this with a company, an Australian company called Medical Devices International. And the thing that's really interesting was that they worked out how to do it for Australia, and they were a nice $3 million small to medium enterprise. Now they thought, wouldn't it be great if we could sell this to the world, because we like to make everyone lose their pain. And the chemistry didn't work when you wanted to scale it up. And that's the bother with a lot of polymer chemistry, is that when you go to try and scale up, it's really, really difficult. So our guys went through, and girls, went through and worked out a chemistry that allows it to be scaled. And so that $3 million company is now a $300 million company and exporting this Penthrox, it's called, or the green whistle, all around the world. And that's something that just happened in 5 years because Australian scientists in CSIRO were able to help them. This is just something where you might have seen it. Whenever you see sports accidents happening, you can see there the different football players and people in, I think, Survivor or something like that. Someone burnt themselves, and it gets used a lot. Just to finish off, we're also coming up with ways to actually be able to detect viruses or detect bacteria, and this is something so that we're able to have very quick detection of something which is potentially going to make us sick. So this is a really lovely idea. This is a clever biosensor on a glove, so that you go and scrape the biosensor on something, click your finger together, and it's linked up with something that goes into your mobile phone, and it'll be able to sense what is present and be able to detect, for example, there's a horrible a nerve agent on a door handle which might have been planted there inappropriately, or it might be something where there's a food, a particular bacteria or something in some food, or on, I think that we saw something there, gym equipment with cold viruses all over it, so that there's a potential to actually be able to have gloves that can then, with a mobile phone, work together to be able to help you be your own, I guess, disease detective. So they're just a couple of things of how we go about using the information that these amazing scientists do, understanding the biology and the way that the viruses and bacteria evolve. And then we're all about trying to make sure that we have ways to solve it, and then also ways to make sure that you can buy it as well. Thanks.
So you've caught my attention with that glove. Who would that be good for? Who could use it? I mean, I can't imagine the public all wearing gloves when they go to the gym or anything like that. Who do you
Apart from
envisage?
hypochondriacs.
Yes, exactly. Which no one wants to look like. How do you see that playing out? Who could use it in the early stages of that technology?
Yeah, that's a good question. I think it'd be particularly useful for things at the beginning, say at places where you're trying to see if there's a, say, pandemic or something like that, and you might be wanting to, at border security, be able to go through and do quick swipes, so that you're, instead of having a test that's got to go away for a couple of days, you're able to do real-time testing. So that's probably the main thing. You have to, though, have incredible sensitivity. So, you know, sort of parts per billion is the sort of thing. So that's very sensitive detectability, and otherwise it's not going to be able to be useful. So that's the critical bit.
And is that the challenge, to get that sort of sensitivity on a glove?
And also a range of different specificities for different things you're testing. So what you do is on the glove there's some nanoparticles there, which are then, they've got surface coatings put on them, and you can change the surface coating depending on what it is you're looking for. So you need to have one glove per thing you're looking for. That's not broad spectrum, it's just looking for one thing.
Fabulous. Okay, so
Watch this space.
Yes, wonderful. So I was just going to ask a couple of questions, because I've got some questions, and then I was going to throw it out to the audience. So I'll just ask a few and then we'll get going. So Sean, I understand you're working on things every day, all day, every day. What's your latest project? What are you working on at the moment?
So I mentioned this survey we've been doing of protein structures, and we've been extending that to all proteins from all organisms. So it's generated 100 million model structures. And I often draw the analogy to the Hubble Space Telescope. So that's sort of a systematic survey of everything that's out there that we can see. And so we're building actually a system that enables scientists to explore this 100 million protein structures, so that whatever they're working in, viruses or bacteria, they can actually see all of the structural information that we have currently very easily in a website. So yeah, it's sort of a Hubble telescope for protein structures, and elucidating the protein structure universe, as we like to call it.
Okay. And so Andy and Rebecca, how would that help you guys? If you can use a website like that, how does data visualization help you guys?
Particularly when we're trying to design new drugs that may be used against viruses, understanding the structure of the virus and the way that it interacts with cells that they use to make copies of themselves can then help us to get a handle on how we might be able to get to intervene between those two to stop that from happening. So that sort of information is critical when we want to design new types of drugs or new types of strategies for dealing with viruses.
And how has that changed what you do?
It's not really changes what we do, but it comes into what we do. So we use multiple different approaches to trying to find solutions to this, and this is one of the approaches that's really becoming popular now. People trying to understand it from that physical three-dimensional look as much as it is from designing the drugs themselves that can sort of fit into those cavities.
Okay. And Rebecca, we were talking earlier about some of the work that you've done, and you were saying that you've detected viruses, but you're not quite sure what they will and won't do. So you've got them sort of there in the lab. Can you tell me a little bit more about that? How you found them, where they are now, what's happening with them?
Well, as I mentioned briefly, I take my bat urine, and basically what I do is I put it on cells in a flask, and try to see if anything happens to the cells. And if the cells start dying or they start joining together and forming these massive cells, maybe that's caused by a virus. And then from there we look at if it is a virus at all, so we're able to sequence what is maybe causing that change in the cells. And then we can go through the virus sequence. We can make a lot of comparison to existing viruses. So are they, do antibodies react to them in the same way, or do they grow in cells the same way? And then what we do to get the best idea of can it cause disease in humans, once we've done as much as we can in cell lines and by looking at the sequence, so on computers, to get the best idea is we put it into animals, and then from there we can see, does it cause disease? What organs does it infect? Can it then get transmitted? There's a lot we can learn from then putting it into animals, and that's where I probably get the most information from. Yeah.
And what sort of timeframes are you working with there? Is it months or years or?
I think each individual experiment doesn't take too long. An animal experiment can take months, and definitely setting up and analysing it takes months. But it's mainly putting it all together that takes a long time. As I said, I've been working on it for 3 years, and I'm sort of got a good understanding of these viruses, but I think there's always more you can understand about them. You can just keep on going for years, really.
Okay, great. I'll just sneak one more question in. Andy, you mentioned the WHO in your talk. They have warned there's a so-called disease X lurking out there that we have no idea about, which is terrifying. Do you have any idea what that might be? Would it be lurking in an animal? What sort of thing are they talking about there? What could we expect from that?
No, I have no idea about it. I guess what they're talking about is that we just don't know. It's the unknown unknowns. It's one of those sorts of things that across time we've seen, outbreaks of diseases that we never saw before. And I think that Beck's sort of given us some really good examples of the diseases that, 20 years ago we had very little knowledge of, and now they're outbreaking diseases, and some of those currently outbreaking at the moment. So we can guess what they may be like, and as we sort of discussed, they could be a particular paramyxoviruses like Hendra or Nipah. We've seen those come out lately. Or they could be coronaviruses like SARS or MERS that we've seen come out. But one really important way that we can keep looking and keep searching and keep up the surveillance around it is by doing what Beck's doing and having a look in, say, bats, who are often the reservoirs for these things, and seeing if we can identify something that may be on the horizon.
Okay. Is anyone keen to ask a question? Yeah? Okay. Have a roving mic. Sorry, I'll get you to grab the mic if you can. Thank you.
I'm just wondering if fruit bats can hold so many viruses without reacting, can we learn something about how to emulate their immune system?
I think that, a lot of people are looking into that, but I think what people are finding is that it's so many different things. It's not just one part of it that's different, so we can target that and use that to improve our response. It's just a combination of everything being different because of millions of years of evolution. Bats are a really old animal order. They split off from other animals about 50 million years ago, so they've had a long time to evolve these special ways of dealing with viruses. And yeah, I think they've definitely identified particular things, but I think that to actually get an effect, we would need to have all these differences.
Just taking us back to Andrew's reference to the 1918 Spanish flu pandemic. Now, I'm not sure whether the panels can illuminate us as to what sort of conditions existed at the time, the end of the Great War, that led to that pandemic that were unique, because I'm not aware of any pandemic before or after, short of the Black Death, that led to such loss of human life. And the other aspect of my question is, what is the likelihood of a pandemic of similar dimensions breaking out today?
So we know a little about the conditions that were around at that point in time, but because it was during a wartime, a lot of information was kept a little bit secret. So, it's called Spanish flu, but it really didn't outbreak in Spain first off. It's just that Spain, because it wasn't a part of the war, announced the numbers of people who were ill from it fairly openly, and therefore it was defined as being Spanish flu. Other countries kept those sorts of information a little bit suppressed because they needed to in that situation. But we do know about it and we see what was there. As I sort of alluded to, what was really intriguing was that different from other flus, this pandemic was mostly directed at people who were mostly healthy and who were mostly middle-aged, rather than say the elderly or children who are quite frequently susceptible to these sorts of things. So it was a little different. Recently, we've had a bit of discussion about how people may have contracted other respiratory illnesses at the same time, so that may have compounded it. But we know that this pandemic had a very different set of responses that people made, and as I sort of pointed out, that overwhelming response that flu can give you with Spanish flu appeared to be really quite a severe response, which led to a lot of the mortalities.
And didn't it also coincide at the time when international travel was beginning to grow, so that then it spread? So you had
so the
a few years
spread of
before
it.
the Titanic happened. So it's you ended up having, and I guess World Wars too meant that you had people moving around the world.
Well, people returning from war, that type of thing too. And again, as I said, it was before a time. All the other pandemics have occurred after the time when we've had vaccines available, which has had quite an impact on the outcomes.
I just pipe in from the sort of structure and sequence side of things. It's a paper just a few years ago that surprised me was they were analysing the genome of that particular virus, and the genomes of these viruses are tiny. There's like 6 to 10 genes. They're absolutely minuscule, and only recently we discovered that there were new genes in there that we'd never seen before. This is only about 3 years ago. So there's a lot of subtleties to viruses that we are really only now unraveling. So this genome, for example, can be read in multiple different ways, and so we think there's only 6 genes, but actually because of different frame shifts and so forth, they can encode more genes than we've previously thought. So I think we're still in the process of even understanding some of the fundamentals of these viruses.
I have another question. With the idea of disease X and not knowing what's to come, Kathy, is it worth scientists, or should scientists be working towards vaccines that treat a range of diseases? Is that possible?
I don't know the answer to that, but I imagine it's probably these people here would answer it. But the thing that's really important is you've got to, the whole process of developing a vaccine or developing a drug is actually very expensive. And so there's always this cost benefit. First of all, who's going to be supporting it? Who's going to pay for all the testing, which is very expensive? It usually goes through multiple trials. Starting off, you've heard about things in animals, and then it goes on to primates, and then it might go on to human trials, and then various ones to show the efficacy. So there's a whole, from taking the idea of having a universal drug that can get everything, I suspect it's unlikely, but I'd be interested to hear what the panel say. But I imagine that you've heard about how the need to have specificity to be able to get in there and have something that can deal with a particular drug or a particular virus or bacteria or whatever is absolutely critical. And if you have a broad spectrum, I think you might end up with a situation too where you end up with resistance building up. We have a bad habit of not taking drugs all the way through to their end, so it actually can lead to also having super viruses or bacterial sort of growing, so the survival of the fittest. Okay. I don't know, the others might have something else to say.
Well, we'd really love to have a universal flu vaccine, which I guess in some respects is a much broader vaccine. Right now, the vaccines that we get are quite specific. And so one that could be a bit more broad-ranging, that could at least give us protection for maybe 5 years rather than just the sort of single season, that would be a great thing to happen. There's a lot of work that's going on now to try and deliver on that. It's been a difficult process because the flu virus is a pretty tough enemy. But we're still working away on trying to deliver that.
Okay, great. Any other questions?
Just wondering, in terms of infectious diseases, how worried we should be as them being used as a tool for biowarfare. And Kathy, in terms of, you know, the glove that you were talking about, is that potentially one way that we might be able to protect ourselves?
Well, first of all, luckily we have a world where there's an agreement that people won't use biological warfare, and hopefully the world will be sensible enough to stick with that, and that's why foreign affairs and good relationships with countries and managing crazy people who think that they'll take over the world and science fiction comes true hasn't happened yet. So let's just hope human good sense continues. But I think one of the things is that, you know, as we see extremism happening, you will have small cells of people who might, you know, develop things, especially with the development of synthetic biology and that sort of thing. That's sort of, you hear about CRISPR technology, which means that you can develop stuff in your garage, and you've got school kids doing experiments where they're able to develop biological materials, and this is where I'm getting way out of my expertise. But the thing is that it's just like the fact that you're getting to a point where it's becoming regular and easy to develop biological things that are able to affect us has a whole question mark over it when you get extremism happening. So being able to detect things is really interesting, and I actually think a glove like that wouldn't be all that useful. There's always a residue. People always have to go to the toilet. People always have to wash their hands. So the best way probably to keep an eye on it is actually to be monitoring our waterways and being able to have things. And you will have seen some work that's been done saying that the big drug capital of the world is actually Perth, a particular suburb in Perth, which has the most number of people taking drugs. How do they know that? Because they actually go through and measure the sewer and look at the concentration of illegal drugs in the sewer. And you can do the same with looking for you know, explosives and for looking for nasties, you know, or precursors for chemical, you know, for biological stuff. So that's probably a better way, is actually keeping that sort of smart pipes monitoring stuff, which is, I understand, the dream of most modern societies, which are trying to work that way so that they can pinpoint it. And also be looking for pollution too, when people doing the wrong thing, pouring stuff down the drain, from us doing the wrong thing and pouring oil down the drain. We might end up with a little fine in the or the police turning up at the doorstep, sort of saying you've done the wrong thing. But the others might have some other views on that too.
Yeah. Anyone else have a question? Here we go.
Thanks for tonight. It's been great. I'm interested to hear your thoughts around whether or not we're limiting our immune system in societal changes. So things like in primary schools, they've stopped being able to blow candles out of birthday cakes and stuff like that, and the spread of germs and diseases.
Who would, anyone? Anyway.
Oh, sorry, to clarify the question, you're saying our immune system has not been challenged enough? Is that, yeah. As someone who grew up out in the sticks in Alice Springs and Darwin, I felt that I think that this is a big thing. And you've probably heard about these people who are giving parasites to, in some cases, to some children who've got autoimmune diseases. In some cases, their immune system has basically been designed to tackle these very strong challenges, and now we're living in this hyper-clean, hypergenic society, and then our immune system is sort of under-challenged, and that potentially explains some of the autoimmune diseases. So I think it's a very real effect as far as I know, but I guess I should really hand over to the experts, so I'm more down on the molecular level. So.
Well, hygiene hypothesis is what they sort of call it. There's a lot of work that's going on to try and understand that sort of situation. You know, when you're younger, if you make a response in a particular way, and how is that memory of that response carried on in your immune system. It's something that there's a lot of really good research being done at the moment in, and I think that in the next few years we'll get a much better idea about how those sorts of things work.
Anyone else? Good.
I've always wondered about that too.
Yeah.
But
Yeah, okay.
It's a bit sad not blowing a candle out, isn't it?
Okay.
A question for Rebecca. If you could be infected with Ebola or Hendra or Nipah, what would your preference be?
Well, good or bad? Really sick or not so sick?
You asked the tough question. Maybe Ebola, because I think that a lot of the time you are able to, not with specific treatments, but just you can manage the infection as long as you have, good care around you and everything. There is a bit more chance of recovery. Also depends on which strain of Ebola, I guess. Maybe I can pick a particular strain of Ebola that doesn't cause disease in humans, which does exist. So maybe I'll go with that.
Is that because you can see the symptoms earlier, that you can get on it earlier, or?
So both Hendra and Nipah virus cause an infection of the brain, and so it's very difficult to, once it's there, it's very difficult to get rid of that infection. Ebola, again, it depends on the strain, but they have found that just managing symptoms can be really beneficial as well. So,
I have another question over here.
Hi, thanks. I'm wondering, in addition to studying the disease, to what extent is helping the process studying individuals who are healthy and resisted the disease or had it and then survived, what can we learn from them, either their immune system or their genome, to complement how we understand how the disease progresses?
I know they definitely get antibodies from people that have recovered from particular illnesses. So I think with Hendra virus, that might be where they got one of the antibodies for it. But then analysing, what antibodies do they have that responded to that infection and potentially fought it off? And then can they grow up those antibodies? Can they produce those antibodies to use as a treatment to then help someone else's immune system fight it off? So they definitely look at it.
So should we be taking these healthy individuals and then sending them into the wild and getting them bitten by bats so that they can be the disease pioneers to bring back not the loot from different civilizations, but the antibodies, which we then harvest and inject into ourselves? I mean, that could be a thing.
I mean, if you can find some volunteers for that, then...
There are probably other ways we could do that.
Yeah.
No, you're spot on with what you say, that by being able to understand how illnesses affect people, it really does help us to develop new strategies for how we're going to deal with those in the future. And as you sort of said before about, when we understand how when you are infected and that you can tolerate that infection, that it doesn't cause you a lot of harm. Some people can live with illnesses, and there are some animals that also, as we said with the bats, they can live with those things. The more we know about that, the more that we can make rational decisions on what type of therapies we may be able to design.
And perhaps just to follow up on that and spell it out, the stochastics, it's been underlying a couple of things we've seen. Viruses evolve incredibly fast. Our immune system evolves incredibly fast as a response, but then at a variable rate across the population. And then, as we referred to with the Spanish flu, the fact that we're joining the world together with modern transportation obviously works against us and in favour of viruses. But then people like the Animal Health Lab are working against that to try to make more targeted approaches. So there's these different effects going on, targeted and stochastic, and the question is which one is going to win.
Hi, thank you very much for the good presentation. It's really amazing to know about so many about virus. And we talked about a lot mainly on the infectious disease. So I wanted to kind of go into something chronic conditions. So as far as I know that some of the virus infections is associated with cancer, like HPV associated with cervical cancer in 75, I think it's 70 to 75% of the cases. And I want to ask the panel what's the latest research in the association with virus and other cancer. Is there any other virus that we should be taking note of in terms of cancer development?
I think that the most exciting thing I've heard in this field is some new experimental technique actually was developed just late last year that could actually image the molecular structure of our entire DNA. I could actually see the shape of it. And the most exciting part of that was they were able to actually dynamically insert viruses into human cells and watch what those viruses did. So we know that they insert their DNA in various places and that causes viral proteins to replicate, but we have never actually been able to see how it actually affects the overall organization. And I think that cancer is driven by the disorganization of the genome. So I think that in some ways, at least for me as a structural biologist, because I think that the answers to many of these questions lie in actually seeing the structure, I think we've sort of just developed some of the techniques, the technologies to actually see that. That's one of my answers. I don't know if anyone else on the panel has others.
There's one of the things that's interesting is you had noticed, Sean, at the Garvan Institute, what they've been doing is a whole lot of studies looking at the human genome and then looking at when people have particular cancers. Why is it that some people will have the same cancer and they get a drug or a treatment and it works, and another it doesn't work? So they've been able to go and look at, it's not exactly relating to viruses, but it's actually looking at the way our bodies react depending on our genetic makeup. And there's things that they've found is that, surprisingly, being able to really help people with very unusual cancers that have been unsolvable by just understanding the way their genome has engaged with that, and then being able to put often a very unusual drug in to cure it and put the person into remission. So there's some really clever things on various levels, looking at how we can use different treatments to be able to treat a range of diseases. And the thing that's really interesting is with diabetes, for example, there's people with a propensity for it, and they found there's a guy who realised, having his genome sequenced, that he could get type 1 diabetes, and it can turn on if you have a very high temperature. And he had the flu, actually, and he was able to monitor his pancreas or whatever the biofactors are that they're able to measure. And by keeping himself in an ice bath, he could actually bring it back down, and he didn't get diabetes. So it's a really interesting benchmark paper that's sort of showing it, sort of almost experimenting on yourself. But it means that we've got a long way to go to really understand how all these things we've heard about tonight all contribute to just the way this amazing human body and the way biology works.
Perhaps a follow-up point, because I work at the Garvan, and this is
that's right.
It's actually very new, but it's not related to viruses, but pancreatic cancer, for example, is one of the few cancers we've made very, very little progress on in terms of the survival rate of people once you're diagnosed. And only in the last few months they've actually been able to change that. But it relies on the ability to do complete genomic sequencing, which is only available in a very small number of institutes, including the Garvan, but just a very few worldwide. So that's an example of, the thing is, we can only cure what we can see, and these technologies like complete genomic sequencing or looking at structures at a certain level of detail provide us this new ability to see. And so it's difficult to predict what's going to happen, but there's some very encouraging results, certainly. And we've actually made some breakthroughs just very recently in pancreatic cancer, which is actually amazing. And some of the technologies hopefully we're developing here will help you further breakthroughs.
Thanks very much. I've had a great time listening to you all. And I'm sure I'm not alone to want to know from Beck how she collects the urine and also whether she's got any favourite or pet bats. Thank you.
Yeah, I get asked this question every time I talk about my project. So what we do, as I said, we don't want to disturb the bats at all. And unfortunately, I wasn't involved in the collection process. That happened before I started. But what we do is you lie down plastic sheets below a bat colony, and then they head off and feed for the night. And when they come back, before they go to sleep, they go to the toilet, and then they can just collect the urine into little tubes and then put that in the freezer. And then I came along 4 or 5 years after they had collected that and was able to isolate viruses from it. And some of our we have samples that go back quite a way, and you can still get live virus just from collecting the urine off a plastic sheet in the sort of early hours of the morning. But I don't have any specific bats that I work on, but I do like the little grey-headed flying fox, which is the picture that I showed. I think they're pretty cute.
I've got a little sneaky question. I've been thinking about this, and is it worth going to all this effort if other countries aren't doing the same?
Well, I would speak a little bit for them, but Hendra, I was actually a little bit shocked that you would rather be infected with Ebola than Hendra, because Hendra is at our doorstep, and I think not enough people realise that, and I'm in awe of the work that these guys are doing to combat it. So,
as far as in my area of finding new viruses, something that's really exciting is that they're trying to launch the Global Virome Project. So they're trying to find all the viruses in all the animals of the world. And they actually are doing a really good job of getting researchers in a huge range of different countries to sample animals, sample sick animals, sample animals in the wild, and try and isolate or identify any new viruses, not even to isolate them, but just to sequence these samples that they've got and see if they can find anything, and try and put together a catalogue of all the viruses that are out there. So that's a good example of, people working together all over the world.
What about you guys?
Oh, well, I think we've got some of the greatest minds in this area, so it's right that even if no one else is working on this, that we do, because we can deliver the solutions to the problems that are out there.
Okay, great. Any other questions? One down here.
I'm just interested when we talk about diseases that are affecting animals, what relationship does that then have to diseases that affect us?
I mean, it depends. Obviously, there are some viruses, let's say, that just infect a particular animal. So they're only found in pigs, or they're only found in cows, and they never leave that animal. But there are a number of viruses that are almost amplified in animals. So in the case of Nipah virus, or let's go with Hendra virus, the Australian example, it goes from bats, but it doesn't, in the case of Hendra, it doesn't go from bats to humans. The reason humans were able to get infected is because it was causing disease in horses. And Nipah virus can go from bats to humans, but it was also, in one case, for a while was amplified in pigs. So it was causing disease in pigs and then moving to humans. So it's really important to keep an eye on what's happening in animals, because there is always that potential, depending on the virus, for it to move into humans. So if we can catch it while it's still in animals, and potentially get a vaccine for animals, which is sort of easier to do, then we don't have to worry about it moving into people then. We can stop it early.
So those types of infections we call zoonotic infections. Zoo because they infect animals and humans. Influenza is a great example of one of those, where primarily it can infect water birds, like ducks, etc., but then transmit across into other animals, other mammals, and even into us in the end. And it's really important that we keep an eye on it and have great surveillance over both animals and what's happening in humans.
Sorry?
Can I ask one more question?
Of course, please do.
Before, when Steph was talking about, if other countries, aren't protecting their own backyards, I mean, is Australia at risk from other countries in Asia, for example, where I understand, some of the flu virus started? Can Australia be affected by bats flying over, or are we at risk in any way there?
Well, we live in a global world now, so Australia's not an isolated island as we used to think of ourselves. So we're always open, I think, to biosecurity incursions that may occur. But one great thing, I think, is that particularly at ARL and with the CSIRO, we work together with other countries that surround us and work with them to try and identify what's going on so that together we can all develop defensive strategies around these types of things.
Is it hard to get approval for animal testing? What's the sort of process behind that?
Yeah, it can. So as I mentioned, I do some work in animals, but from the point of saying I would like to do an animal experiment to actually being able to do an animal experiment can take months. You have to apply to an animal ethics team, and they really make sure that you're treating the animals as well as possible. So you fill out a multiple page report, you say how the animals are going to be fed, how they're going to have enrichment activities, how they make sure they've got a friend with them at all times. Like, they really want to make sure that we're treating the animals well. And that animal ethics committee is made up of researchers, it's made up of members of the community, external researchers, so not just at our facility. And you need to work with these people to actually design the experiment to make sure you're going to get your results using as fewer animals as possible.
I think that's right. I think we're as concerned as most people are, and we really believe in that philosophy of replace, reduce, refine. And so wherever possible, we look to replace the animals in these experiments. It's critically important.
I would like to know which has been the most exciting day in your career. So that you feel like, oh my God, this is
That's a
worth
fabulous
it.
question. You go first. Most most exciting day in your career.
Most exciting day, wow.
Putting them on the spot.
Yeah, it's pretty good.
I think for me, there's many days. It's usually when I think about something, an experiment, and I say, I guess this is going to happen. You know, I look at the conditions, I say, I think this will happen. And then when people come back to me and say, it worked, it was like this, and I always go, yes, I knew I was right. So that really pumps you up. You know, it's great to have ideas about inventions, and then to see those inventions come to life. That's an amazing feeling, a great experience that you have. And look, the whole team really contributes that way, and we all really love it when that happens.
Yeah, it's often if an experiment has gone wrong for a very long time, and I've then spent months trying to fix the experiment, and I think, this is it, it's going to work, I've made all these changes, and then it does finally work, and the results are exciting. That's always very good.
I think for me, I've started organising scientific meetings, and just for me personally, I mean, I enjoy my own work, and that's very, very exciting. But to get 200 of the brightest minds in the world together in a room, focused on one topic, and the first time I did that, that was very exciting, because that's like accelerating what I can do, and yeah, that was fun.
I think for me, because a lot of my stuff is incremental, and it's not like I have that eureka moment so often. It's I think a little bit like keep working away at something. But probably one of the most exciting times is when I realised that my own personal research is in superconducting electronics, which can be used for biological applications, but also for mineral exploration. And we developed a system that could be used for looking for deep ore bodies, and we commercialised it, and suddenly realised that it actually had found a whole lot of mineral deposits which were mined in a way that actually helped Australia through the financial crisis. So ore bodies which my little device found actually were mined, and their shipments of nickel sulfide actually helped Western Australia be financial during the global financial crisis. And when someone put that together and told me, did you realise you saved the Australian economy? I was pretty chuffed about that. So.
Well done.
Yeah, thank you.
Thank you. A bit extreme, but I thought it was pretty exciting.
Here we go.
Hi, so I heard that the flu virus is so dangerous because it mutates really quickly. So other viruses, what's their sort of mutation rate, and what makes the flu so special?
I think they all mutate quickly. The issue is with flu is about the rate of transfer from person to person. But
with flu, it's a different kind of virus from other viruses. Quite frequently, they have what they call proofreading. So when their genetics is being maintained, if that mistake comes up, it proofreads it and fixes it up. But with flu, it doesn't work in that way. It allows those changes to occur. And so that's why it's a little bit different. It does have a higher rate of mutation because of those. It doesn't have that proofreading ability to do that. It works to its advantage sometimes, and it works to its disadvantage in other circumstances.
Okay. Is there a microphone for Eamon?
Question for Andy: Is a hot whiskey good for the flu?
For medicinal purposes only. That's right.
Okay. Kathy, I was interested in what you thought about this. I read an article with the chair of the US-based Global Health Council, Jonathan Quick, saying the world has a cycle of panic and complacency when it comes to infectious diseases. And he says the cycle is currently in complacency. Do you see that with the work that you're doing?
Oh, absolutely. I think Australia's in a very complacent thinking in general because we've had unprecedented economic growth and lots of money flowing around in the country, even though I know we don't all feel that. And many researchers think we don't get as much money as we'd like to do the work we could do. Nevertheless, it's something where, as a country in particular, we've got fantastic healthcare, we've got fantastic schools and universities, and a very stable government system. Even though we think they've been changing political parties regularly, it's still pretty middle of the road, and so it's not like we've gone from fascist to communist and things like that. So it's I think Australia has been the lucky country in the sense that we've got to a point where we expect it and we think that everything will just be fine, and which is probably a good chance it will be, but only because a whole lot of people are sort of paddling fast underneath when we're gliding along and like duck legs. And I think we need to be aware that things are fine because a lot of people behind the scenes are actually keeping it fine.
And does anything need to change there, do you think?
Well, yeah, sure. I think a lot of people don't realise really basic things like if I bring this thing from overseas, it's only one. No one's going to suffer from that. If I bring my little pet bug which I picked up, or that grain or that flour or something, or that meat product from overseas, I'll just bring it in, and they think that somehow that they have no impact when they do. Another one is, I think someone was saying, stay at home when you have the flu. I think, I'm sick, but it won't matter because somehow my flu or my cold isn't going to spread. And so, there's this thing that somehow I'm special and what I do won't affect everyone else, and I think that's pretty naive thinking.
Oh, we've got a question at the back.
I have a question for the panel about Renzane, the flu medication. Is there a limit of when you can start having it? Or can you take it any time during the flu?
Oh, this is for antivirals?
Yes.
Yeah, apparently they only work really after in the first 24 hours of major symptoms. If you take it after that, it's too far gone, as far as I know. So it might reduce it a bit, but apparently you've got to get it really fast. So if you feel the symptoms coming on, they do recommend you go to a medical practitioner quickly and get an antiviral as quickly as possible, because they can really suppress the symptoms of flu and reduce the impact it has on you, which is why they stockpile it for pandemics.
Sean, you were saying earlier that data visualisation is actually quite a competitive arena. So how does Australia hold its own, and how do you hold your own in that world?
Well, I think it's data visualisation has always been there. We've always been looking at data, but I think the recognition of the importance of it has only come recently, really with the sort of the complexity of the data we're doing, with the new technologies that are being developed. And so, yeah, this concept of data visualisation applied to the life sciences, to medical sciences, and to things like viral research, I think it's very new, actually. And the techniques that are being developed are also very new. And so, yeah, we're lucky that we have actually pretty world-leading expertise that we've managed to cobble together. It's a little bit like what Kathy was saying, and we actually have really quite a leading edge in that. So my team, we're running one of the biggest conferences in that field, so That's really a privilege, and we get to invite some of the top people in the field.
Okay, great. Questions in the back?
Hi. I just wanted to ask each of you, in your field, what do you reckon the next big thing would be?
Kathy, you go first.
Oh, I'll go first. Next big thing. I think the next big thing is actually going to be seeing how digital stuff actually, and our understanding of the engagement of digital things into the way our brains work. I think our brains are really not understood very well. They're amazing supercomputers, I guess. And they also deteriorate with old age. And we're going to have a whole lot of us when we get old, where we're going to have a level of brain decay. So I think the next big thing is actually understanding how the brain works, but also either how we can retard the decay of it, or we can augment it by putting electronics and stuff into our brain. I think that's, for me, where the next big thing is going to be. So I'm looking forward to, that Harry Potter pensieve where you're able to put your thoughts in. That's, being able to download that and then store your memory and then being able to put it back after you've had a brain rewire. I can't wait, yes.
Me too.
Actually, I think the next big thing is going to be that someone gives us a solution to flu vaccine manufacture so that we don't have the problems that we have today with saying, we're worried about it running out.
I'm not sure about the next big thing, but I know something that I would really like for my research is to just be able to look at this virus, the virus sequence, and say, yes, that's going to be able to cause disease in humans, or no, that's fine, you can stop looking at it now. It would be nice to be able to get that response really early instead of investigating it for years, maybe.
Well, William Gibson said the future's already here, it's just not evenly distributed. And I think I've seen the next big thing, and I think it's encapsulated in a book by an Australian, Nielsen, called Reinventing Discovery, and that's the idea that as communities of scientists worldwide will begin to collaborate on specific things. It could be an infectious outbreak. If you think about it, we've got expertise around the world, around Australia, and if an outbreak happens somewhere, if we could get the right minds together, focused on the same data, on the same problem at the same time, and talking to each other, we could really reinvent how we discover, how we advance. We're already doing that to some extent, and some of us, I'm trying, like other people are trying to build tools to facilitate that. So we have it to a little bit, but I think we can have it a lot more, and we can have it sooner than we can have brain implants. Google, in a sense, is already a small part of that, but we're talking about a Google specifically for science and for specific areas of science, and I think that's going to be really powerful.
And how could you make that happen sooner rather than later?
You could fund people who are building those kind of things. I didn't want to get into having a little bitch session about funding, but I think there are people around the world who are already pioneering this, and I think you really have to look at what has worked. Google is an outstanding example, but again, it's too generic, and there are tools, and it's in fact Australian who really articulated this vision, this Reinventing Discovery book. I think we've simply got to. It's a question of where we put our focus and where our attention, and that involves also money.
Okay, great. Any other questions out there? So it goes to economic cost. Kathy, this is probably another one for you as well. Sickness versus research dollars, probably Andy and Rebecca, all of you. Obviously, sickness has an economic cost. Maybe this goes to the complacency idea as well. Is there enough being spent on dealing with these things and protecting ourselves to stop that economic cost of sickness and infectious disease outbreak?
Well, you've just asked a great question for government employees who aren't meant to comment on that. Nevertheless, look, Australia actually invests a huge amount of money, Australian governments, into research and development. The thing that isn't as great is industry doesn't invest as much compared to other countries. And so that's why we always look a bit lopsided with our funding. But probably one of the biggest things is prioritisation. We actually have to work out what is the most important thing to invest our research dollars in. Where are we able to make the biggest impact, particularly as having swelling of groups like specialist facilities like we have at ARL, and the ability to have visualisation, but also making the most of the multidisciplinary interactions so that Australia's pretty, it's sort of interesting because we're a big country landmass-wise, but with a very small Vegemite scraping of population. I mean, the population of Australia is the same as Shanghai city, so and the landmass is the same as mainland USA. So it sort of puts it in perspective. So we have the advantage of actually having a point where most researchers actually know each other to some extent, and organisations like CSIRO, where people tend to work there a long time and get to know each other across boundaries, so that you are able to have those interactions and do things at the boundaries, which is where I think really interesting things are happening. And I suspect where the future is for us is actually making the most of what we have, working together, prioritising, and showing that the stuff we choose to do will have the impact we need to make Australia great, so to speak, in a global context.
Quick follow-up, how important are international companies in supporting that sort of research? How much is private support? I get the impression that maybe Australian companies aren't quite there, but are international companies?
Okay, so Australia is the land of the small to medium enterprise. Okay. 97% of companies in Australia are small SMEs. But the thing is that most of them are S's, not M's. So most companies are about less than 20 people. And that's part of our job, to try and make them grow and be globally competitive. But the thing that's sort of interesting is that if you're a small company, you often don't have the ability to invest. So you decide, do I take my family on a holiday, or do I actually pay for some research from CSIRO or university? And it's really interesting to see. You might have seen the announcement a couple of years ago by the federal government, of the National Innovation Statement and Innovation and Science Agenda, NISA, and that actually has had an enormous cultural change in Australia. I think what we're seeing is that combined with CSIRO's ON program, which is trying to create entrepreneurialism, saying, can we take this research and turn it into something which can be commercialised, is seeing a rapid change in the way companies are realising that they can invest in science and technology and be able to be globally competitive and be a better company, be more successful in delivering a product or an outcome or a service or something. So I think that that Australia is in a changing situation. We've also got a whole lot of interesting things happening where, believe it or not, if you look at most countries in the world that are got big dollars being spent in research, it's usually from a defence and defence perspective. And you might have seen a whole lot of discussion about defence expenditure, like submarines and frigates and all that, and they're trying to grow defence industry, with our Minister for Defence Industry talking about this. The reason they're doing that is that not only does it make Australia sort of safer on one level and having a sovereignty to be able to do that, but the other is that it's a sort of a backdoor way of boosting the economy and getting big companies to actually invest in Australia. And so you're beginning to see that now, and it's sort of a complex ecosystem of all this stuff coming together. And you'll see, some of those things like investing in even defence industries, are actually also in a biological way as well as all the materials and all the other things like that. There's environmental issues, for example, with the Great Barrier Reef being a huge question mark for us. Can we actually save it as global warming happens, or climate change happens? And what we're seeing is companies like Boeing, which is a major international multinational, which we all fly in their jets, that they are very interested in how they can contribute to the research in understanding what's happening there, because they feel that they contribute to it from a sort of government, an organisational responsibility, as well as tourism, ecosystems being healthy, so we've got a healthy country. So I think we're in a state of flux and change, and it's going to be pretty exciting to see where we land.
Okay. Question?
Thank you all. It's been very interesting tonight. But how willing are scientists around the world to share the results of their research? Are they more protective of their intellectual property than they used to be in the past, given the commercialisation that's surrounded the research these days?
I think the scientific community is pretty good at sharing data. You know, that's one of the good things about the community that we have, that publications of our information and our data are one of the key things that we use. If intellectual property is protected, you know, usually with a patent, etc., then people have no problems in sharing them because the patent protects that. And I think that these days most people, if they are wanting to commercialise their data, move fairly quickly to get that patent in place and then start sharing the information. So it's pretty good.
I think you'd be surprised at the percentage of world knowledge that's available right now online. It's a shockingly large percentage. Certainly in the life sciences, it is the default to make data openly available, and often very quickly after it's been acquired.
Do we have any last questions? We might wrap it up there. Okay, well, thank you all so much for coming. It's just been fascinating, a fascinating discussion. Thank you to Sean O'Donoghue, Rebecca Johnson, Andrew Bean, and Kathy Foley. Thank you so much for your wealth of information and for sharing it with us. And thank you guys.